Orientation program for NEET aspirants. NEET தேர்வு எழுதும் மாணவர்களுக்கான இது ஒரு வழிகாட்டி நிகழ்ச்சி. இன்றைய வகுப்புல மல்டிபிள் சாய்ஸ் கொஸ்டின் டைப்ஸ் பிராக்டிஸ் பத்தி பார்க்க போறோம். Now we come to that urine formation. You know very well in urine formation there are three stages. One is ultrafiltration and tubular reabsorption, and third one is tubular segregation. Then ultrafiltration occurs in the glomerulus. You know what is glomerulus? This is a network of capillaries which is formed in the Malpighian corpuscles, right? Where filtration takes place. For filtration we need a force. That force, you know what are the force available here? The difference in the sizes of the afferent arterioles and efferent arterioles creates the force for filtration. Here the glomerular hydrostatic pressure is 55 millimeter of mercury, which is opposed by two forces. One is the colloidal osmotic pressure, which is about 30 millimeter of mercury, and another one is capsular hydrostatic pressure, which is 15 millimeter of mercury, together 45. So 55 minus 45 remaining 10 millimeter of mercury is the net filtration pressure is responsible for ultrafiltration in the glomerulus. This is very important part. And another interesting point you have to remember, the glomerular filtration rate is about 100 to 125 ml per minute. So the volume of filtrate formed in 24 hours is 180 liters of filtrate. Out of this 180 liter, nearly 1.5 liter is formed as a urine. The remaining 178.5 liters, that constitute nearly 95% of the glomerular filtrate, get reabsorbed. So you must know only 1.5 liters of urine is formed out of 180 liters. Then next we come to that renal or tubular reabsorption. You know, usually the essential substance like glucose and amino acids and some ions, potassium ions, calcium ions, these are all pigments, and these are all reabsorbed in the proximal converter tubule. That essential substances are all absorbed in the proximal converter tubules, and then the remaining things enters into the Henle's loop. In the Henle's loop, you know, U-shaped tube, Henle's loop, it consists of two limbs. One is the ascending limb, another one is the descending limb, another one is the ascending limb. The descending limb is permeable to water and impermeable to salts, whereas the ascending limb is impermeable to water but permeable to solutes like sodium and chloride and some salts. Now we come to that distal converter tubules. The distal converter tubule where the tubular segregation takes place. You know, mainly segregation of hydrogen ions and potassium ions and ammonium takes place in the distal converter tubules, and from the distal converter tubules it enters into the collecting duct where water is get absorbed. As the end, the urine form in the collecting duct is hypertonic to the body fluid concentration. That is, urine is twice or thrice more concentrated than our body fluid concentration. Okay, so you know, my dear student, which one is the correct sequence of urine formation? You know, there are three steps in urine formation. One is the filtration, ultrafiltration. Second one is the reabsorption. Third one is segregation. So the answer is B: filtration, reabsorption, and segregation. These are the three important sequences in the urine formation. Next, this is a very interesting question. Ornithine cycle or urea cycle or Hensley cycle. This cycle shows how urea is formed in the liver. You know, my dear student, first the cycle starts with the formation of carbamyl phosphate, which is formed by the combination of two molecules of ammonia and one molecule of carbon dioxide. They combines to form carbamyl phosphate. This carbamyl phosphate combines with ornithine to form citrulline. The citrulline combines with aspartic acid to form arginosuccinate. This arginosuccinate gives fumaric acid and produce arginine, right? This arginine, the enzyme arginase, which is present in the liver, with the help of the enzyme arginase, arginine hydrolyzes to form urea and ornithine. So the cycle starts with ornithine and end with ornithine. That's why this is called the ornithine cycle. In this ornithine cycle, there are three amino acids. One is ornithine, another one is citrulline, another one is arginine. In this the enzyme arginase, which is present in the liver, play an important role, which helps for hydrolysis of arginine into urea and ornithine. What is the, now we come to the question part. What is the correct sequence in the ornithine cycle of urea formation? My dear student, this cycle starts with the ornithine. When this ornithine combines with carbamyl phosphate, it produces citrulline. And the citrulline combines with aspartic acid, forms arginosuccinic acid. And this arginosuccinic acid gives rise to arginine. And this arginine with the help of arginase is converted into urea and ornithine. So the cycle starts with ornithine, first step, 1 ornithine, 2 citrulline, 3 arginosuccinic acid, 4 arginine, 5th one is urea. So select the correct answer using the code given below. The option is 2. So first ornithine 1, second one is citrulline 5, arginosuccinic acid 3, arginine 2 and urea 4. So option B is the correct answer. Now we come to that human vertebral column. When the newly born child have 33 vertebrae, whereas when they grow into an adult, some vertebrae fuse, and as a result, the adult human vertebral column consists of 26 vertebrae. Then first cervical spine consists of 7 vertebrae. Cervical vertebrae வந்து மொத்தம் 7, கழுத்து எலும்புகள் மொத்தம் 7, and மார்பு எலும்புகள் thoracic spine சொல்லுவோம், அதில் there are 12 thoracic vertebrae, and then இடுப்பு எலும்புகள் lumbar 5 vertebrae, and sacrum, all the 5 vertebrae unite to form single vertebrae, sacrum, and similarly 4 tail vertebrae unite to form a single coccyx. So totally 26 vertebra in the human vertebral column. So which one of the following is showing the correct sequential order of vertebrae in the vertebral column of human being? My dear student, the vertebral column starts with cervical. Cervical is followed by thoracic, and thoracic is followed by lumbar. Lumbar is followed by sacral and coccygeal. The answer option is fourth one. Fourth one is the correct sequence. Okay, my dear student, now we come to that very interesting part, that is how muscle contraction takes place. The impulses or the signal for the muscle contraction was initiated in the central nervous system. Now this signal carried by the motor neuron to the sarcolemma of the muscle fiber. So the junction between the motor neuron and the muscle fiber is called neuromuscular junction, or you can say motor end plate அப்படினு நீங்க சொல்லலாம். See, once the signal reaches the neuromuscular junction, you know what happens, and action potential is develops. You know, you know what happens there. The neurotransmitter, namely the acetylcholine, is released at the junction. This acetylcholine creates an action potential in the neuromuscular junction. This action potential enters into the sarcoplasmic reticulum, which releases calcium ions, and these calcium ions bind with the troponin, you know, troponin of the actin filament, thereby which removes the masking of active sites for myosin. Now the myosin is set free. Now the myosin head, which contains an enzyme ATPase, which you know, enzyme ATPase, in presence of calcium and magnesium ions, you know what happens, it splits or break down the ATP into ADP, and energy is released. With the help of this energy, this myosin filament bind with the actin filament. When it bind with an actin filament, it pulls the actin filament towards the A band. As a result, the Z zone moves towards the center, and as a result the muscle starts to shortening, and this is what we call it as sarcoplasmic contraction. This is explained by a famous theory called sliding mechanism or sliding filament theory. According to this theory, the thin filament that slide over the thick filament, and this results the shortening of the muscle fiber or sarcoplasmic contraction. Arrange the following flow chart in the correct sequence of events. Okay, see, you know very well the muscle contraction is best explained by sliding filament theory. This theory states that the contraction of muscles is due to sliding of thin filament over thick filament, right? Okay, so the first step in this muscle contraction is the impulse from the CNS, or you can say the signal for muscle contraction was initiated by the central nervous system. Then as a result, what happens, that impulses were carried to the neuromuscular junction. What is neuromuscular junction? The junction between the motor neuron and muscle fiber is called a neuromuscular junction. When it reaches the neuromuscular junction, what happens? The synaptic vesicles of the motor neuron release a neurotransmitter called acetylcholine. This acetylcholine creates action potential in the sarcolemma, and this action potential release calcium ions from the sarcoplasmic reticulum into the sarcoplasm, and as a result these calcium ions bind with troponin on actin filaments, and this as a result the active sites of actin are exposed. Then the myosin head now binds to the active sites to form cross bridges, and these actin filaments are pulled towards the H zone, and as a result, you know, the H zone disappears, and this is what we call it as shortening of sarcomere or muscle contraction. Now we come to the question part. Arrange the following flow chart in the correct sequence of events. So the correct answer is D, E, G, A, B, I, C, J, H, K, F. This is the correct sequential order in which the events for muscle contraction takes place. Now we come to the meninges. Meninges are commonly called brain membranes. You know, the brain is protected by three membranes. The outer membrane is the dura mater. which is thick dura matter, and a middle arachnoid matter, and inner pia matter, which closely surrounds the brain. So you must know the brain membranes are called meninges. Dura matter, arachnoid matter, pia matter. Dura matter is outer, and middle arachnoid matter, and inner pia matter. The infection of the meninges called meningitis, or we say commonly called brain fever. Meningitis. Whenever the brain membranes are affected by virus or bacteria, then it is called a meningitis. The correct sequence of meninges from inner to outer side. Here also the question பார்க்கும்போது உங்களுக்கு மிக முக்கியமான அந்த வார்த்தை எதுனா inner to outer. அது தெரிஞ்சாதான் நம்மளால கரெக்ட் ஆன்சரை நம்ம செலக்ட் பண்ண முடியும். இன்னர்ல இருக்கிறது நம்மளுக்கு நல்லாவே தெரியும், பையா மேட்டர், arachnoid matter, dura matter. இதுதான் outerல இருக்கு. So the correct option is C. Now we come to that cranial nerves in human beings. Is a very important topic. You must know, in case of man there are 12 pairs of cranial nerves. You must know all the cranial nerves in sequence. That is very important. The first cranial nerve is olfactory nerve. The second cranial nerve is the optic nerve, and the third one is oculomotor, and fourth one is tracheal, and fifth one is trigeminal, and sixth one on the abducens, seventh cranial nerve on the facial, eighth on the auditory, ninth glossopharyngeal, 10th vagus, 11th spinal accessory, and 12th hypoglossal. So you have to know the cranial nerves in order. There are 12 pairs of cranial nerves. You should know the names of the cranial nerves in order. அதில் அப்புறம் பிறகு அதை வந்து எது பியூர்லி சென்சரி, எதெல்லாம் பியூர்லி மோட்டாரு, எதெல்லாம் மிக்ஸ்டு, இப்படிலாம் நீங்கள் அதை கிளாஸிஃபை பண்ணி படிக்கணும். ஸோ இந்த தலைப்பில் நம்ம அந்த சீக்வன்ஸை மட்டும் நம்ம தெரிஞ்சுக்கிறோம். There are 12 pairs of cranial nerves in the order of olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, auditory, glossopharyngeal, vagus, spinal accessory, and hypoglossal. Okay, so once again, based on that sequence, you come to the question part. Which one of the following is the correct sequence of cranial nerves in chordates? You know, இதில் எல்லாத்துலேயுமே நீங்கள் பார்க்குறீங்க. ஃபர்ஸ்ட் வந்து abducens. Abducens வந்து உங்களுக்கு தெரியும் 6th cranial nerve. So abducens followed by facial 7th, auditory 8th, glossopharyngeal 9th. So the correct sequence of cranial nerves given in that option on the C. C is the correct answer. Now we come to that human ear. You know, the human ear, there are 3 parts: outer ear, middle ear, and inner ear. The outer ear consists of the pinna. You know, the pinna which collects the sound waves and passes to the through external auditory canal. And when the sound waves hits the tympanic membrane, which is the inner part of the external ear, tympanic membrane, and which vibrates. When this vibration is carried by 3 bones, small bones in the middle ear, they are called malleus, incus, stapes. அது கூட நீங்க ஆர்டரா தெரிஞ்சுக்கணும். அது ஈஸியா ஞாபகம் வச்சுக்கலாம். மிஸ் malleus, incus, stapes. And from the stapes, the vibrations carried to the inner ear. Inner earல வந்து 2 மிக முக்கியமான பார்ட் இருக்கு. ஒன்னு semicircular canal, which helps for balancing. Another one is cochlea, which is auditory. The auditory part of the ear எதுன்னு கேட்டா cochlea ன்னு சொல்லலாம். From the cochlea, the auditory nerve emerges out, and which carries the sound waves, that is sound waves to the brain, which detects the sound waves. Okay, so how the sound waves are transmitted through the inner ear? There are 3 parts in the ear: outer ear, middle ear, inner ear. Outer ear la pinna irukku, external auditory canal, and tympanic membrane. And இப்போ வைப்ரேஷன் tympanic membrane ல ஸ்ட்ரைக் ஆன உடனே வைப்ரேஷன் ஆகும். அந்த வைப்ரேஷனை மிடில் இயரில் இருக்கிற மேலியஸ் இன்கஸ் ஸ்டாப்ஸ் வந்து கண்டக்ட் பண்ணும். தென் இட்ஸ் கேரிட் டு த இன்னர் இயர். இன்னர் இயரில் காக்லியாவும் செமிசர்க்குலர் கெனால் இருக்குது. செமிசர்க்குலர் கெனால் வந்து பேலன்சிங் இன் ஃபங்க்ஷனு. காக்லியர் இஸ் த ஆடிட்டரி பார்ட்டு. அங்கே காக்லியர்லேருந்து auditory nerve carries the impulses to the brain. Okay, now we come to the question part. Identify the correct sequence of organs or regions in the organization of human ear as the auditory mechanoreceptor organ. நம்மளுக்கு தெரியும் வெளியில இருக்கிறது வந்து பின்னா. பின்னாவுக்கு அடுத்தது auditory canal. Auditory canal is followed by tympanic membrane. Tympanic membrane இருக்கிறது அதுக்கு followed by middle earல இருக்கிற ossicles, malleus, incus, stapes, அப்புறம் inner earல இருக்கிற cochlea. அந்த cochlea இருந்து auditory nerves emerge ஆகுது which carries the impulses to the brain. The correct option is 1. Okay, now we come to that retina of the human eye. You know, my dear student, retina is the sensitive part of the eye, right? And which consists of three various kinds of cells. The first one is the photoreceptor or visual receptors, and the second one is the nerve cells, bipolar nerve cells, and then amocrine cells, and then ganglion cells. You know, photoreceptors, there are two types of receptor. One is a rod, another one is cone. One rod, இன்னொன்று cone. Cones for color, rod for twilight vision சொல்லுவாங்க, vision, that is for light. Here rod is consists of a protein called a rhodopsin, whereas the cones contains different types of substance, colored pigments like synopsin or idopsin or porphyropsin. சொல்லிட்டு there are different types of pigment. Porphyropsin for red color, idopsin for green color, and synopsin for blue colored light. So cones la three types of pigments irukku. Rods la only one type of protein, that is called a rhodopsin. Then next we come to the bipolar neuron. You know, very interesting. இந்த கேள்வி இப்படி கூட கேட்கலாம், பைபோலார் நியூரான் எங்க இருக்குன்னு சொல்லிட்டு ரெட்டினா ஆஃப் த ஐன்னு சொல்லுவோம். பைபோலார் நியூரான் சொன்னா it has two process. One acts as axon, another one is dendron, right? So they carries the impulses from the photoreceptors to the other part. The amocrine cells which connects ganglions with bipolar cells with ganglion cells. You know, ganglions are group of nerve fibers which contains cyton of various nerve cells. No, ganglion are groups of nerve fibers. From the ganglion, the impulse is carried by the optic nerve to the brain. So retina of the human eye. Now we come to the question part. The order of the three layers of cells in the retina of the human eye from inside to outside. மறுபடியும் இந்த கொஸ்டின்ல கீ வேர்டு பாருங்க, inside to outside is. உள்ள இருந்து வெளியில, inside to outside. இன்னர்ல நம்மளுக்கு என்ன செல்ஸ் இருக்கும்? கேங்லியன் செல்ஸ் இருக்கும். கேங்லியன் செல்ஸ்க்கு மேல பைபோலார் செல்ஸ் இருக்கும். பைபோலார் செல்ஸ்க்கு மேல போட்டோ ரிசெப்டார் இருக்கும். ஸோ ஃப்ரம் இன்சைட் டு அவுட் சைட் சொன்னா கேங்லியன், பைபோலார் அண்ட் போட்டோ ரிசெப்டார் செல்ஸ். ஸோ த ஆப்ஷன் சி இஸ் த கரெக்ட் ஆன்சர். Now we come to that meiotic preface. You know, my dear student, meiosis is a reduction division, right? It consists of four phase. One is a preface, metaphase, anaphase and a telophase. அதில் meiotic preface வந்து ஒரு long phase which consists of 5 substages. அதில் வந்து 5 stages இருக்கு. First stage வந்து leptotin, அப்புறம் zygotin, மூணாவது packetine, நாலாவது diplotin and then diakinesis. இந்த leptotin-உடைய characteristic features என்ன அப்படின்னு பார்த்தீங்கன்னா, the chromatin reticulum condensed into chromosomes. So during leptotin, chromosomes are visible. Then second stage பார்த்தீங்கன்னா zygotin. Zygotin, you know, zygotin where the pairing of homologous chromosomes takes place. Homologous chromosome na similar chromosome, One is from the father, the other one is from the mother. Homologous chromosomes. இதை நம்ம pairing of homologous chromosomes technically என்ன சொல்லுவோம்னா synapsis or synapsis அப்படின்னு நம்ம சொல்லுவோம். The next one வந்து Packetin stage. Packetin stage is a very important stage because during which crossing over takes place between non-sister chromatids. What is crossing over? Exchange of genetic materials between two non-sister homologous chromosomes, and that is very important event which is responsible for recombination of genes or which is responsible for variation. So Packetin is an important substage during meiotic preface, right? Crossing over. The next step, diplotin. During this diplotin, what happened? The dissolution of synaptonemal complex takes place, and then diakinesis, where is characterized by terminalization, that is the movement of chiasmata towards the end, that is terminalization takes place. So first leptotin la chromosome becomes visible, zygotin la synapsis nadakudhu, packetin la crossing over nadakudhu, diplotin la synaptonemal complex dissolution ஆகுது, diakinesis la terminalization process நடக்குது. Now we come to that question part. Which of the following is the correct sequence of meiotic prophase 1? Meiotic prophase 1 la enna sequence la nadakudhu paathom. Just now we studied about the 5 substages. And the substage la first substage edunu sonna leptotin. Leptotin ungalku theriyum. Leptotin is followed by zygotin. Zygotin is followed by packetin, and packetin is followed by diplotin and diakinesis. Option C is the correct answer. Leptotin, zygotin, packetin, diplotin and diakinesis. So the option C is the correct. See, based on the same concept, another interesting question. Arrange the following events in the meiosis in correct sequence. இப்போ நம்ம just now we studied about the various events that takes place in the meiotic preface. அதில் நம்ம பார்த்தோம் first step என்ன அப்படின்னு பார்த்தோம்னா இன்னும் zygotinல என்ன நடக்குதுன்னா synapsis நடக்குது. So synapsis is the first answer, which is followed by crossing over. Crossing over நடக்குது. Crossing overக்கு அடுத்தது என்ன நடக்குதுன்னா disjunction of chromosome. Disjunctionனா என்னன்னா separation of chromosome. The separation of chromosome நடக்குது. That is followed by dissolution of synaptonemal complex. That is followed by terminalization of chiasmata. So the correct sequence வந்து synapsis. So option 3 is the correct answer. First one synapsis C, followed by B, followed by D, followed by E and followed by A. So option 3 is the correct answer. இது வரைக்கும் நம்ம மல்டிபிள் சாய்ஸ் கொஸ்டின்ஸ்ல சீக்வன்ஸ் டைப்ஸ் எப்படி தயார் பண்றதுன்றத பத்தி பார்த்தோம். நம்ம படிக்கும் போதே அந்த தலைப்புகள்ல அமைந்திருக்கிற இந்த சீக்வன்ஸ் டைப் கொஸ்டின்ஸ் எல்லாம் நம்ம தேர்ந்தெடுத்து அதுக்கு முக்கியத்துவம் கொடுத்து படிக்கும்போது குறைந்த நேரத்தில் நம்மளால விடையளிக்க முடியும். தேங்க்யூ.